Today we're talking about Chapter 6, Section 5, and this is all about Punnett squares and probability as we use them in genetics. Punnett squares are simply ways for us to illustrate possibilities resulting from a genetic cross. It is a grid system, and it predicts all of the possible genotypes from that cross. The axes, or what we write across the top and down the side, those represent the possible gametes that a parent could pass down. And the boxes inside the Punnett square show us the possible genotypes of their offspring. So this diagram is handy because it's color-coded. The green is parent 1. They're heterozygous, so that means they can either pass down a capital A or a lowercase a. Parent 2 is colored in purple. Same thing, they're heterozygous, so they can either pass down a dominant or recessive allele. So inside of these boxes, we see the 4 possible combinations of the offspring. So purple means that allele came from parent 2, green means it came from parent 1, and you can see all 4 possible combinations. 2 of them are essentially going to be the same, the heterozygous ones, but notice the difference between these 2 is which allele came from which parent. In this one, the dominant allele came from parent 2, but in this box over here, the dominant allele came from parent 1. So that's why we have 2 different ways of representing heterozygous in this Punnett square. And what we get from a Punnett square is most often a ratio. We can look at the genotype ratio or the phenotype ratio. This obviously is written as genotypes, but we can interpret that as a phenotype. The 3 boxes that you see here that have at least one capital A allele would show the dominant phenotype, so this would be a 3-to-1 phenotype ratio. But if we are focusing on genotypes, there's 1 homozygous to 2 heterozygous to 1 homozygous recessive. So we would say the genotype ratio is 1 to 2 to 1. Now that we just looked at is called a monohybrid cross because it only involved 1 gene. So if we are looking at one specific characteristic of an organism and how it's inherited, a monohybrid cross or a simple 4-box Punnett square is what we would use. We just discussed that genotype ratio. We saw it in the example on the last screen, a 1 to 2 to 1 ratio. Whenever you write the ratios, you always start with the number that is the most dominant, so 2 dominant alleles would be the most dominant alleles that we have in this particular example. And then 1 dominant, 1 recessive, that would be 2. And then 2 recessive alleles is the 3rd number that we have here, giving us a 1 to 2 to 1 genotype ratio from a monohybrid cross. The phenotype ratio, as we discussed on the previous screen, is 3 to 1. So if we're talking about purple flowers being dominant over white flowers, you would expect these individuals from the first 2, so the 2 dominant alleles or 1 dominant allele, 1 recessive allele, doesn't matter what the genotype is, all 3 of these plants would have purple flowers. So our phenotype ratio is 3 to 1. And here's our Punnett square that shows us that to be true. This Punnett square shows you both genotypes, which would be the Fs, and the phenotypes, which would be the color of the flower presented. Now, a dihybrid cross is a cross that involves 2 genes because di means 2. So in a dihybrid cross, we don't really focus on the genotype ratio because there are just so many possibilities of different genotypes. But this ratio, the phenotypic ratio of a dihybrid cross, is something that will be very helpful for you to recognize. A 9 to 3 to 3 to 1 phenotypic ratio is what you would expect if you have 2 heterozygous parents and you're crossing them looking at 2 specific genes. And Mendel did this when he did dihybrid crosses to learn about the law of independent assortment, which is, if you remember, when the alleles separate independently for different genes during the process of meiosis. So here's an example of a dihybrid cross. Notice, first of all, this is more than just 4 boxes because we have to track 2 different genes at the same time. We're looking at the color of the seed as well as the shape of this seed. So this is a yellow round seed. Yellow is the dominant allele, and if we look at the shape, round is dominant over this wrinkled recessive allele here. So notice that the parents are hybrids, right? It's a dihybrid cross. So if you have 2 heterozygous parents, we have to figure out what gametes will each parent make. Now, normally, if we were just looking at capital Y, lowercase y, that's easy. You put a capital Y here, lowercase y here, move on. But we have to know what are all of the possible combinations of Y and R that this parent can produce. So the best way to do that is if you think about that FOIL method from math. F for first, take the first Y and the first R. That's one possibility. Capital Y, capital R. O for outside. Take the outside Y and the outside R. You get a capital Y, lowercase r. And that's our second possibility. I for inside. Our inside Y and our inside R would be lowercase y, capital R. Here's the third possible gamete. And finally, last, take the last Y and the last R, and you get both of them as being lowercase. So that would be recessive Y, recessive R. All 4 of those gametes are possible from a parent who is heterozygous. So these 4 gametes are what we are writing across the top and what we're writing down the side. So whenever you do a dihybrid cross, you have to make sure that the gametes that you're writing have 2 letters. Whatever gene you're looking at, you need 1 letter from each gene. So we have a gene Y and a gene R in this example. So you need 1 Y and 1 R. We wouldn't have 2 Ys here because remember the law of segregation tells us that those 2 Ys separate from each other into different gametes. That's why we never have 2 Ys in any of these gametes, and we never have 2 Rs. There's always 1 Y with 1 R in each of these gametes. And the easiest way to figure out what those are to follow that FOIL method, just like what you learned in math class. Now, sometimes a Punnett square could be difficult. We get more and more complex problems. You would have to draw a larger and larger Punnett square. So probability is going to become your friend when you're calculating the likelihood of something happening and it's more complex, maybe with 3 genes or 4 genes. The probability is just the likelihood that something would happen. It's not a guarantee. It just tells us how likely it is for something to occur. And it's going to predict an average number of occurrences, not an exact number. So, for instance, if we have a 75% chance of the offspring of the pea plant being purple, that doesn't mean it always has to be exactly 75% of those offspring have purple flowers. Maybe it's 77%, or maybe it's 73%. So probabilities are predictions, but it's not the absolute rule. To calculate the probability of anything, you probably know this again from math class, you take the number of ways that event can occur and you divide it by the total possible number of outcomes. So this helps us in genetics because it applies to random events, and during the process of sexual reproduction, that fertilization process is essentially random. It is not predetermined which sperm cell is going to get to that egg cell first. So we can kind of think of a Punnett square as a coin toss. If we think of this coin as a gene and there's the head allele and the tail allele, there's equal probability if we were to flip a coin that it would land on heads versus tails. So the Punnett square is already doing a lot of our probability calculations for us if we have it set up with 1 allele here, and then 1 allele on the side and the other allele down on the bottom. So that is how our probability rules relate to those Punnett squares. Now, a rule of multiplication is going to come in handy when we start to get into the more complex genetics calculations. And the rule of multiplication allows us to calculate the chance that an independent event or two different independent events will occur simultaneously, meaning at the same time. If that is what you're calculating, you are going to multiply their individual probabilities. So let's look at a simple example, one that you can probably do off the top of your head, and that is a cross between a heterozygous purple flowered plant and a homozygous recessive white flowered plant. We want to know what is the probability of producing homozygous recessive offspring. Now, you would normally just make a really simple Punnett square for this, and you can figure it out very quickly from that. But we're going to use this simple example to show you how the rule of multiplication works so that you can then use it to apply to more complex calculations. So first of all, the first event that we need to happen is we need the heterozygous parent to pass down a recessive allele, because the two simultaneous events are going to be inheriting a recessive allele from the first parent and inheriting a recessive allele from the second parent. So the chance of parent 1, who's heterozygous, passing down a recessive allele is 1/2. They have one recessive allele that they can pass down out of two possible alleles that could be passed down. Only one of them is recessive, so 1/2 is our probability. Now, if you look at the second parent, they are already homozygous recessive. That means they only have recessive alleles to pass down. So they have a 100% chance of passing down that recessive allele. It would be 1/1, technically 2/2. There's two recessive alleles over two possibilities, but that simplifies to 1/1, or just 1. So that's going to give us, according to the multiplication rule, 1/2 times 1 equals 1/2, or a 50% chance that we're going to get a homozygous recessive offspring from this cross of those two parents. So let's check that with the Punnett square. And again, this is easy to do probably in your head. Let's take a look at the parents. We have them both listed there. Here are their potential offspring. And so we can see of these offspring, 2 of them are homozygous recessive out of 4 possibilities. 2 out of 4 is 50%. So this checks out. Our second rule of probability is the rule of addition. And this happens when you're calculating the chance of 2 or more events occurring, but they could occur separately, meaning you can have one thing or another thing. If that's the case, they don't have to happen at the same time. If they can happen separately from each other, we would add their individual probabilities. So an example here that is, again, a very simple example that you can do easily with Punnett squares is, what is the probability of heterozygous crossed with heterozygous parents producing homozygous offspring? Now remember, there's 2 ways you can be homozygous. You can either be capital F, or lowercase f, lowercase f. So we have 2 different ways that we could satisfy the requirements of the question. And they don't have to happen simultaneously. That means we can take the probability of each one separately and add them together. So the chance of inheriting a dominant allele from the first parent and a dominant allele from the second. We know that there's a 1 in 2 chance of this parent passing down a dominant allele. Again, that's because they have 1 dominant allele to pass down out of 2. And same thing for the second parent, a 1 in 2 chance of passing down their dominant allele. So that's 1/4. Then we're going to look at the chance of inheriting the recessive allele from the first parent and the second. So again, a 1 in 2 chance of the recessive allele being passed down from parent 1, a 1 in 2 chance of that recessive allele being passed down from parent 2, and we multiply those together because those would happen simultaneously, and we get 1/4. Add those together and we get a 50% chance. So there is a 50% chance of these two parents that are both heterozygous producing homozygous offspring, because we have to consider either homozygous dominant or homozygous recessive. Now we can check this with a Punnett square, and again, I know this is a simple enough example to have done initially with the Punnett square. So let's see how this works. We've got our two heterozygous parents. We fill in the Punnett square, and what we're looking for are homozygous offspring, so homozygous dominant or homozygous recessive. There they are. 2 out of our 4 boxes meet that criteria. So if we add those two together, we get 1/2, and that's our 50% possibility. So we have now used the rule of addition and the rule of multiplication together to solve this problem. The rule of multiplication to figure out the chance of each parent passing down a certain set of alleles, and then we added those together using the rule of addition because those two events did not have to occur simultaneously, and that gave us our final probability of 50%. Now, you may be wondering, why in the world are we doing that when we could just draw a Punnett square? Well, consider this question. You might see a question similar to this on a quiz or a test. And if you were to try to draw a Punnett square for this question, it would be really, really large because we are now looking at 3 different genes: flower color, seed color, and seed shape. So if you are trying to figure out what percentage of the offspring from this cross would have purple flowers and green seeds and a wrinkled shape, then it becomes much easier to use the laws of probability. So if we made a big tri-hybrid Punnett square for this, it would be 8 by 8. That's 64 boxes. Nobody wants to spend all the time filling in 64 boxes of a Punnett square. So instead, we do this the simple way by considering each gene individually. We can look at just the flower color gene. If we are self-pollinating this pea plant and it's heterozygous, that's 2 heterozygous parents, capital F, lowercase f. We can easily make a Punnett square for that, and it looks like this that you see right here. Our second gene for seed color, we can also make a Punnett square for that. And then finally, our seed shape, we can make a Punnett square for that. Now we want offspring that meet all 3 of these criteria simultaneously, so we would need to multiply the probability of each one happening. So what's the probability of getting offspring with purple flowers? Multiplied by the probability of getting offspring with green seeds, multiplied by the probability of getting wrinkled seeds. So if you look at each Punnett square, it becomes very easy to figure out those individual probabilities. There's a 3 out of 4 chance of getting purple flowers, a 1 in 4 chance of green seeds, and a 1 in 4 chance of wrinkled seeds. So if you multiply those 3 together, because again, we need them to happen simultaneously, we get 3/4 times 1/4 times 1/4, and that gives us 3 out of 64, or about a 4.7% chance. So we have now very easily figured out the answer to a question that if we had tried to draw the Punnett square for this question, we would probably still be drawing those boxes. So this is why the rules of probability are helpful for you in understanding genetics. We will be doing further practice with this in class, but if you have any questions and this is not making sense to you yet, please make sure you ask those questions so that you can move forward and do the practice.